From Microbe TV, this is Immune, episode number 9, recorded on June 20th, 2018. I'm Vincent Racaniello, and you are listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome. Immuners? Do we say immuners? Have we decided what we call ourselves yet?
Whatever you want.
Welcome back for another episode.
Just don't call me late for dinner. That's how it goes, right?
I think so.
I've never, in my adult life, I've never been told to come to dinner.
Oh, I thought you were going to say in your adult life you've never been late.
Oh yeah, I've been late. I try not to be, but not late for dinner because we never had dinner together. Everyone always has their own dinner.
Yeah,
it's the modern family, right?
That's right.
Also joining us from Worcester, Ohio, Steph Langel.
Hey there. Yep, things are going well here in Worcester. It's like 77 degrees, pretty humid. It was really hot last week, into the mid 90s. But
2 days ago, we had almost 100. It was very, very hot.
Yeah. Was it humid too?
It was unbearable.
I hate to complain, but I'm not. I mean, I like the cold, but we'll always complain about weather because it's a common
What do you
thing.
want to call us?
Immuners? Immuners. Our listeners have come up with some pretty good things, I think, throughout our emails. I'll have to peruse them and see if I can find. Immuners is good. That's fine.
You can, you can let them write in for a few more months and see what happens.
Yeah, maybe we'll have a contest
There's
and give away.
One over on TWiP. We have one of my favorites is Twipanosomes.
That's great.
Isn't that a good one? Before we go into our paper today, I wanted to. I was over at iTunes today. I'm looking at other science podcasts. We have TWiV number 500 next week, and I was going to.
I know. I heard that.
Right. I was looking to see how many other science podcasts have gone 500 or more episodes, and there aren't many. But I tell you, there are a lot of science podcasts out there. There's a lot of competition for ears. Anyway, I went to Immune on iTunes to see the ratings because we have 29 ratings. We have 27 5-star ratings, 1 4-star rating, and 1 1-star rating.
Oh no! Maybe they were confused. Maybe they
most people like it, except there's one person who is Bucky62738 says this podcast is very one-sided when they discuss politics. Not every scientist is a liberal and has your one-sided ideology. Please stop talking about politics and keep to science behind stuff. After all, you don't have degrees in economics and political science. To which I would like to respond. When politics impinges science, we have to talk about it.
I agree.
It doesn't matter that we don't have political science or economic degrees. That's just a reason for you to say not to talk about it, frankly. But if the president is cutting the NIH budget, we will talk about it. If the president is saying vaccines are bad, we will talk about it, right? I think it's our job. There is no separating politics and science, right?
And the other thing is we're not just putting opinions out there. This is backed up. The effects of cutting NIH funding is directly on research, which is what we're supporting.
For sure.
And I'm a proponent of vaccines. If there are policies that come out against vaccination and we believe based on our scientific knowledge and the evidence that's out there that this is the right thing to do, we need to speak up.
Yeah, it's not about being liberal or not. It's just about the right thing to do.
That's correct. It's not whether it's left side or right side. It's whether we believe based on the scientific evidence and our experience that it's right or wrong.
Yeah.
Yeah. And it's interesting. There are a lot of topics that are hot button topics that I have seen, from both sides. So if you consider GMOs for one, both conservative, liberal, there's people who they're misinformed across the board. Vaccines, same thing. So we're going to talk about it. And for that particular listener, the combined amount of time we maybe mention something is like such a small percent. If you can't handle it, you're so worked up. Fast forward. We got good stuff here. You'll be fine.
So I noticed I went over to Meet the Microbiologist, which is a interview style podcast by Julie Wolf. She's at ASM. And Bucky had a comment there. And this is great.
Bucky seems upset.
Bucky says, there is no annoying chatter or talk about the weather.
Ah.
That's what he said on that about that podcast?
Yeah. Yeah.
So he's upset about politics and weather.
And the weather, which we talk about mostly. We don't do too much here, but on TWiV we do. But some people just get cranky, you know? And here we are. We're doing this for your pleasure and learning, right? We're not doing it to make money or to advance ourselves, or it's because we love science. So it's just funny when you get that kind of criticism. But that's fine. It has to just flow off of our backs as if we were ducks, right?
That's
quack,
right.
quack.
Something like that. That's how it goes. On TWiV, I often get emails about politics as well. And but I can't. I think the science and politics are intertwined because our money, NIH money, comes from the government. It's taxpayer money that's appropriated by the president and Congress. So it's intertwined. There's no way around it. And even what Einstein said, science and politics cannot be separated.
That's right. There is a famous quote by him, yeah. Do they complain about the weather chatter on TWiV?
Some people do. Some people don't like it, yeah. But, you know, the funny thing is that many people, when they write, they'll go, hello, the weather here in Chicago is, right?
But, you know, it's an icebreaker. It's something that you expect. It's kind of a transition of whatever else you're doing going into that.
I did have a suggestion, and maybe we could implement it as our weather update. But somebody asked, like, they're not in science. They said, well, what do you do all day? You know, what this week are you up to? So we could say today, you know, I don't know.
Yeah,
people like the stories of what we do.
What are you doing today, Steph? Are you meeting pigs? Meeting pigs today?
We do have, we bled baby piglets this morning. We spun down the serum, and I'll be running antibody ELISAs tomorrow. So that's what
How about you, Cindy?
Oh, I was actually doing experiments. I have a new rotation student who's fabulous. And we were staining some cells, teaching him how to do immunofluorescent microscopy. So we were looking at cells under the microscope. And that's one of my favorite things. You know, when you do a staining and you put it under the microscope and you turn the fluorescence on and things light up and you can visually see where things are and how they work. I think it's fabulous. It's so fascinating. So I still get excited about that when I do things in the lab.
Yeah, I changed the CO2 in a liquid nitrogen cylinder this morning. That's about my extent.
That's so exciting.
Lab. And then I spent the rest of the morning arranging guests for future podcasts, like next week in Texas and all the details. And the following week, ASV, I'm trying to arrange our immune at ASV. And I'm thinking this morning, I've almost had it with this arrangement crap. It's tiring. I just want to talk about science. I need a tour manager.
You need somebody.
I need a manager to do all this stuff.
I'll say,
go get Cindy Leifer for TWiV. Go email her, tell her, and figure out a date so I don't have to do with it. You know, that kind of thing?
Yep.
And, you know, I was just at...
It's minor, but it takes a lot of time going back and forth.
And this is not... I mean, I just did ASM, a couple of podcasts there. I had to arrange those ahead of time. One of them, someone canceled at the last minute, so I had to dig up guests at the last minute while I was in Europe for another podcast. That's not really what I want to be doing, but I guess if I want to do these, I have to. Which makes me think, recently I'm thinking, do I need to do all these podcasts?
Don't drop this one.
No, I'm not going to. This is brand new. I won't drop it. But there's some,
Good.
You know, there's some others. Well, I guess you could say they're so infrequent that it doesn't matter, but I don't know. I'm having a little self-introspection today. That's what I'm doing.
So here's the thing. Here's the challenge for our listeners. Call in or email in and let Vincent know how important this is and give him the energy to keep doing this.
Or go to Patreon and give money so he can hire somebody so he doesn't feel this way.
So
that would be great.
This is the
then
bottom
we can
line.
bring more to you.
I wish I could hire someone. And in fact, the most important thing to me is that I would like these shows to go on for a long time beyond me, right? So I have to put in place some kind of organization that's independent of any one person, right? And we don't have that in place right now. I mean, if I disappeared tomorrow, all the podcasts would stop. because nobody can figure out how to do them, right?
It would take a little while, yeah.
It would, yeah.
I mean, you guys could probably say, "Oh, let's record this, figure out how to do it," but you wouldn't know how to get onto the website, right? All that stuff, and that's not good. So I gotta put something together.
So are there philanthropies or small grants that one could apply to that would fund something related to science communication that might be useful? Maybe our listeners might know.
I have applied for a few in the past. I've inquired of Howard Hughes. Actually wrote someone at the Howard Hughes, an education director, and they said, what you're doing is great, but we want to do our own thing. Okay, thank you. I wrote there's a foundation here in New York City that funds a lot of communication. They said they only want to do traditional media like radio and film. I've tried NSF. So now my thing is I'm going after donors, people who would have money, and say, okay, here's a 5-year plan. We need, I don't know, let's say $1 million to hire a couple of people to run the business and make it sustainable in 5 years. Because I think if we could build our audience out a little more, we could then get sufficient advertising to sustain it. But I can't do that myself. I need someone to do it.
That's right.
Unless I decide to quit, my job, but I really enjoy it, so I don't want to quit it at the moment. So at some point I will, but anyway, that's all. Not really science, but it's the backstory here.
Right.
We do have a real science story for you today, which was spurred by a visit I made, I don't know, a month or two ago to Emory University in Atlanta. And I met with Max Cooper, and he told me all about lampreys and their immune system. Now, both of you tell me, Max told me he discovered B cells. Is that right?
In the literature, it does say that.
So,
although
yeah.
I think now, was he the one though originally in chickens who discovered the IgY? Because I think that was the original discovery.
I don't know, but
it may be.
I had no reason to doubt him. He's like 85 years old. He still has a big lab. I sat in his office with him, and he told me all about Lamprey variable lymphocyte receptors, and I said we have to do this someday on immune. So today we're going to talk about this.
Yeah, I had invited him a few years back here to Cornell, and because I had heard about these when I was a postdoc, and I just thought it was the most fascinating thing in comparative immunology, and I just loved it, and I followed it. And when he came here, he was telling his story about the B-like cells and the T-like cells, which we'll get into in the lampreys. And it's such an interesting convergent evolution. It's more than one way to solve a problem of being able to recognize microbial infection and antigens, and I just think it's so cool.
Yeah, definitely.
And he came to my seminar, in fact, sat in the front row and promptly fell asleep.
Nope.
Which old people tend to do.
Do you know when I was at NIH, there was an individual who I'll leave unnamed for now, but if people know of this, they might realize this. It was somebody who would sit in the front row and look like they were asleep the entire time, and then when the speaker finished, they would raise their hand and ask the single most insightful question you could ever imagine. And you're like, how do they do that?
I've heard of this. I have heard of this. Maybe they're absorbing in their sleep. I don't know.
I guess.
Do you think they're inherently pranksters and they just like to kind of put you off your game, and they think it's fun?
Well, some people like to just close their eyes, right?
Right.
That way they're not distracted.
Take all that time to
listen.
Make them slide.
It could be. It could be. I have trouble staying awake, especially when I'm running around a lot. And my lab person, Amy, was here in my office. He was telling me about some sequencing, and I fell asleep right next to her. And she said, "Are you okay?" I said, "Yeah, it's a little boring what you're telling me." I had asked her, "How much is it going to cost to sequence? 50. We have got 50 trachea from wild New York City mice.
Oh wow!
And we want to see what viruses are in them." So I asked her how much it would cost, and it wasn't really that interesting to know how much it would cost.
Well, I guess at least you're honest.
Yeah, I mean, if it's really interesting, I'll stay awake. But I don't know. I have trouble staying awake, and even in seminars, if it's not so interesting, I'm just tired.
When I'm really tired, I'll stand.
I think that's a good
yeah,
idea.
that's a good.
I'll stand in the back.
You can't fall asleep that way, right?
Well, if you do, it'll be
You
very
fall over.
You fall over.
That's great. All right. All right. Let's talk about lampreys. First of all, I have a quote from the paper, which I'll tell you about in a moment. This is a wonderful way to start. Quote, immunoglobulins are a crown jewel of jawed vertebrate evolution. Through recombination and mutation of small numbers of genes, immunoglobulins can specifically recognize a vast variety of natural and, I would say, human-made organic molecules. I think that's just a wonderful summary of antibodies, which we've talked about twice on Immune, number 3 and number 6.
So antibodies must be important this is our 9th episode, and we're talking about it for the third time. Now, wait a minute. 3, 6, 9, and there's 3 of us. I think all 3 of those are you, Vincent, wow. Right?
Probably because in each case it had something to do with viruses.
Viruses, yep.
I think one was antibody-dependent enhancement.
Yep.
And the second was the bifunctional antibodies, right? Where you have 2 specificities. Yep. And today, Lamprey antibodies we're going to talk about. So that little statement, jawed vertebrates. This is a group of animals called the Gnathostomata. Jawed vertebrates. There are 60,000 species, things like cartilaginous fish, bony fish, amphibians, reptiles, birds, and mammals. And all of these animals, jawed vertebrates, have T lymphocytes, they have B lymphocytes, and they use VDJ recombination to create diversity in both T-cell receptors and B-cell receptors. I think we've touched on that a little bit before, right, in immune?
Yeah, we have a little bit, yep. But the idea is generally that you have cassettes in the genome that you pick up one cassette and then you put it next to another cassette and you put them together in a specific order to make a gene. So the TCR and BCR, T-cell receptor and B-cell receptor, are the only genes in the genome that are not encoded in a functional gene. So they have to be physically recombined in order to make a functional gene, which is really interesting because you're talking about messing with the DNA and cutting it and pasting it together in different ways, which we don't normally think of that being something that cells should be doing. But it's important, critical, and is normal in a T cell and a B cell, and only in a T cell and a B cell.
Now, if you go back 550 million years, I wish I could do that because it would be so interesting.
It would be.
That is when the jawless vertebrates diverged from the jawed vertebrates. So we have vertebrates that have jaws and that don't have jaws. And there are not many jawless vertebrates around. There's still some around today. Remember, there are 60,000 jawed vertebrates, about 100 jawless, and these are lampreys and hagfish.
Now, I don't remember what hagfish look like, but lampreys are they look like a leech, a big leech, right?
Yeah.
And they have a mouth that has a whole bunch of little teeth. It's like a round mouth with a whole bunch of little teeth, and they grab onto the side of something and just
hagfish are like eel-like.
Things out.
They're long things.
So they're the same then.
And you might have seen the hagfish. They have this ability to produce a huge amount of slime. Yeah. And I don't know under what conditions, if they're a defensive. But if you look up images, you can see huge amounts of slime. Both of these animals are they're very bizarre looking. And you think there are only 2? Do you think maybe there's others we haven't found in the depths of the ocean?
I don't know.
Could be.
Maybe.
I mean, one should assume we haven't found everything out there, right?
Yeah, right.
I mean, we didn't even know there were bacteria in the ocean up until 20 years ago, right?
Yeah. Oh, yeah. Look at all that slime. Oh, that's crazy. Right. Oh, yeah. And these are the funny thing. We were saying this before we started. I showed my son pictures of these, and he commented that he thought it looked like the worm from Dune. So if you know those sandworms with those big mouths that are round and they kind of, have all the teeth, that's what these things look like. But you should definitely Google what they look like because they're very interesting.
So these are jawless vertebrates, and some time ago people started studying them to figure out, comparative studies, how they're different. And they were found to mount immune responses. Lampreys in particular could, for example, they would reject a graft. You could inject them with viruses and bacteria. They would mount an immune response. But as soon as we started sequencing their genomes, we found they didn't have T-cell receptors, B-cell receptors, or even MHC.
That's right.
So what is going on here? So people started looking for genes of the lamprey whose expression is upregulated in lymphocytes that were stimulated with an antigen of some kind. And they found a lot of sequences encoding what turned out to be many leucine-rich repeat modules. So these are stretches of leucines repeated over and over again, and there were many different ones. Each module was very different, and the number of modules was different, but they always flanked with some invariant sequences.
And this is very reminiscent of what a toll-like receptor looks like in our system.
So when I was talking to Cooper, he's telling me this, and I said, oh my gosh, it's a toll-like receptor, because if you look at the pictures of toll-like receptors, they're leucine-rich repeats. They form these beta strands, and they make a nice, cool thing, right?
And they had the conserved caps on either end. Yep. But those are static, right? TLRs are encoded in the genome, and the sequences of them do not change with the exception of small polymorphisms.
Right.
So there's something different about these.
Yeah, so these are lots of variable modules, flanking sequences invariant, and all these LRR leucine-rich repeats seem to have derived from a single gene lacking most of the coding region for it. So although nearby, so if you cloned a cDNA from an mRNA, that leucine-rich repeat came from a single gene, but there are many others in the genome as well. So somehow specific ones were getting into each message. So basically what was found was that in these lymphocytes of the lampreys, adjacent leucine-rich repeat modules are incorporated by recombination, which generates the diversity. And so people started thinking, well, maybe this is an antigen receptor. One lymphocyte makes one assembled variable lymphocyte receptor gene is what it was called, VLR. It could almost be VRR, except L. Variable lymphocyte receptor gene. So they're clonally produced by a single lymphocyte. And eventually they showed that these molecules are the antigen receptors. They will bind antigens.
Right. And when you say clonally, we're talking about each individual lymphocyte expresses one and only one of these. Right. So they each have an independent specificity. And when you put a whole group of them together they can recognize a lot of different things, but each one recognizes one thing. And that's how our immune system works with T cells and B cells.
And both lampreys and hagfish eventually were shown to have these. They have multiple. I think in the paper you'll see they work with VLR-B, but there's also VLR-A and C. So these are membrane-bound proteins, the variable lymphocyte receptors. They have an N-terminal cap, and they have multiple leucine-rich repeat modules. There's a connecting peptide and then a cap at the C-terminus, and then a stalk that connects it to the cell. So they're present on the surface of what are called T-like lymphocytes. And these develop, it's really cool if you look this up, in what looks like a thymus, but it's at the tip of the gill filaments in the fish.
It's crazy.
It's amazing, crazy stuff, right? So these proteins are eventually secreted, of course, right? And they have a curved structure that looks very much like a toll-like receptor. If you just look up toll-like receptor and look at the pictures, you'll see it's a curved structure with lots of leucine-rich repeats, and these VLRs look very similar. And a number of structures of VLRs bound to antigens have been solved by structural methods, and you can see that the antigens bind in the curved or the concave part of the molecule.
And which is different from where it would bind, like for instance on an immunoglobulin, because they're complementary determining region loops. They bind flatter. It's not a concave structure. So I think that'll be interesting when we talk into the paper about the similarities between what they bind, but yet they have this different structure.
So toll-like receptors can bind nucleic acids or proteins. Cindy, in that case, does the ligand also bind in the concave region?
So it's really interesting because each TLR seems to bind its ligand in a different way. So typically 2 of these TLRs come together, and if you think of them as Cs, so these curved solenoid Cs, they'll go sort of facing opposite directions and come touch each other. So the 2 opposite facing Cs. Some of the ligands bind at the top where the 2 come together. Some bind on the side. Some bind where the 2 come together, like on the face where they touch each other, the 2 TLRs, and some bind in the concave part of it. But it's not like you just have if you take the palm of your hand and you form a C, it's not like every ligand just sticks there in the palm. So they all bind in very different ways. There's I'd have to look it up, but there's a Cell paper review article that shows really nicely all the different ways that they can bind. But it's not like a one size fits all for them. It's unusual how these TLRs work and why there's so many different things. And the other thing is that some of the TLRs can bind more than one ligand, and they bind them differently. So there isn't a conserved way that a TLR binds a ligand.
So is it thought that the TLRs evolved from these VLR-Bs or VLRs, or were the TLR-like molecules first and then these others evolved from them?
So I think it's an example of using the same idea in different ways. So a convergent evolution, more than one way to solve a problem. Just because, so leucine-rich repeat is not a structural component that's unique to a TLR or a VLR. It's present in some other proteins. And so somehow that structure cassette was useful to solve both this problem to bind antigen-like things in the VLRs and to bind structural microbial components in the TLRs. And so it's weird. So the VLRs are sort of like a T or a B cell receptor and sort of like a TLR. So like a B or T cell receptor in the fact that they use this cassette method and they recombine to make a functional gene. And like a T and B cell receptor where they bind, the ligand in one general similar way. But like a T cell, similar to a TLR, they are using these cassettes in a way to recognize microbial structures as well.
Right.
So I don't think that it's a case of one derived from the other, but I'm not sure what people have done way back looking at, where are these separated 550 million years ago to ask whether one came from the other. But I think that it's just that the cassette was used in both and then used differently. I think there's an example also in, what are those little spiky animals that are in the water that are like a round, what are they called?
Sea urchins?
Sea urchins. So I think sea urchins have like hundreds and hundreds of TLRs, and that's their only innate mechanism.
Oh, wow.
So that's another way to solve the problem, to duplicate a functional full-length TLR many, many, many times in the genome to get more the ability to recognize more than one thing.
Right.
So I guess there's different ways to solve that same need to recognize many, many different things with a limited repertoire of receptors.
Now, these VLRs are multivalent.
Yes.
There are 4 or 5 identical chains linked at one end. You can almost think of an IgM molecule, right? A multimeric IgM, where the antigen binding regions are at the outside, and then the inside they have these stalk regions. They're actually linked by disulfide bonds. So this gives you like 8 or 10 antigen binding sites, so you have lots, you have pretty high avidity. Remember, immunoglobulins, you have a heavy and light chain, and that's 2 heavies and 2 light chains with 3 CDRs, complementarity determining regions that give you the antigen binding site. So I guess
2 of them, one on each end.
Right.
Yep. Right.
Now, IgM has 5 times 2 would be 10, right? They're 5 pentameric IgM, right? Which you make early on. In an immune response. The idea is you haven't yet matured the antibody by maturation in the lymph nodes, so you make this multivalent IgM, so you get a little better avidity before you do somatic hypermutation.
Right. And when we talk about avidity, we're talking about not changing the affinity, which is the strength with which the binding region binds to the antigen. But the fact that you can bind more than one of those increases the, it's avidity, but it increases the strength of the interaction. I don't know, what would we use as an example for that? As if you were able to grab onto something in two places versus one place, you'd have a stronger grip, even though your grip in each hand isn't necessarily different.
Yeah.
Right.
A little bit on how these molecules are made. As the lymphocytes develop, there is a VLR gene, and a sequence in the middle of it is replaced with these LRR sequences, and that gives you the diversity. So you have a whole bunch of LRRs that are put in to form a single VLR. Now, if it were an antibody or an immunoglobulin, it would be done by a recombination activating gene, right? But there's no RAG in these individuals.
Right.
So rather, it is done by a process called gene conversion, where the LRR cassettes are copied into the final gene from where they reside in the genome. And this is an interesting process that needs an enzyme called an activation-induced cytosine deaminase, AID, which plays a role in somatic hypermutation of antibodies in the jawed vertebrates. But there's no somatic hypermutation here. It's really interesting. There's an AID involved, but it's needed for gene conversion, not somatic hypermutation. But it doesn't matter because all of the LRRs in the genome of these lampreys can give rise to combinatorial diversity of over 10 to the 14th specificities. You could have that many different antigens recognized by these VLRs. It's really amazing, right?
It is. It is.
So they don't need somatic hypermutation. So that's the whole thing in summary. You have jawed and jawless vertebrates. They have very different receptors to recognize antigens. You know, the details are different, the way immunoglobulins versus VLRs are made and how they diversify. But in the end, it's kind of a similar thing, right? You have as Cynthia said, there's no one gene in the germline. It has to be put together, and it just happens to be two different kinds of proteins that are assembled to do this. Really amazing. It's cool. So studying, old things like these lampreys can be really illuminating. One last comment. If you look at the sequences of these jawed and jawless vertebrates, a common ancestor seemed to have had both VLR-like and T-cell receptor, B-cell receptor-like molecules. Okay, so they seem to be both present in ancestors, and one went to the jawed vertebrate lineage, and that became antibodies, immunoglobulins, and RAG was needed for that, and then the other became the jawless vertebrates. It's pretty cool.
Right. Yeah. The last thing that I thought was cool when I was talking with Max about this, the idea these VLRs come in two flavors, and you sort of mentioned that the VLR-A and the VLR-B, and one of them looks more like a T-cell receptor and that it's surface bound exclusively and has some mechanism to recognize whatever it's recognizing by maybe a phagocytic-like cell. But then the other one acts more like a B-cell receptor where it can be surface bound or secreted. So it looks more like an antibody. So I just thought that was really fascinating that it also developed this idea that you can have the surface bound or the secreted versions of these, and it's clonal to each depending on which VLR gene it expresses. So I thought that was really interesting too.
Yep. Cool. All right. So this is all meant as background for a paper, which I came across a while ago. It's actually suggested to us on TWiV by one of the authors, John Yewdell, and it's published in eLife. It's called Lamprey VLR-B Response to Influenza Virus Supports Universal Rules of Immunogenicity and Antigenicity. And these are from two groups at the NIAID of NIH and Emory University, Altman, Bennick, Yewdell, and Heron. And in this paper, they basically want to know if we immunize lampreys with a well-known antigen, i.e. influenza virus, do we get the same kinds of responses? Do we direct it against the same parts of the proteins, and do they have similar functions? So what they do here is they get lamprey larvae, and I have a picture that we'll put in the show notes of a person's palm with tiny lamprey larvae. Okay, they're not going to bite you yet because they don't have that big grinding thing at one end. But these are purchased from commercial fishermen, and they bought them. They're just outbred lampreys, and they immunize them with influenza virus, and then they collect the blood of these lampreys to characterize the VLRs.
Now, did they inject them or did they incubate them in the water? Because for some fish immunizations, you can actually just put the infectious agent or whatever into the water and it can immunize them.
I think they injected them, if I remember correctly. All right. They injected them, in fact, 3 times.
And I wonder, I mean, we know that in our species and other species around us that the route of immunization does matter in determining immunodominance. So maybe if it was in the water, it would be more of this mucosal based response instead of what they're looking for is systemic in the blood. Well,
so I just found it. They immunized them 3 times by intracelemic cavity injections.
Mm-hmm.
Okay.
So.
All right. So they use a H1N1 influenza virus, the subtype of influenza based on the surface glycoproteins. And this is a 1934 isolate called PR8, Puerto Rico 8, and it's commonly used in the laboratory. They inject this into the lampreys, and then they can simply run out the protein on a gel, and you can see the increase in the VLR-B, which they said goes up 7-fold in the serum, which is amazing, right? Because if you did this in a mouse, it wouldn't go up.
Yeah, Cindy, what do you think about that? I was taught that, our response to things is not really different between species, meaning like the dose response normalized to how big the animal is. There's not this huge influx of immunoglobulins after activation. So it seems like the lamprey have an ability to increase it way more than humans or mice.
Yeah, we get elevations in specific antibody responses, right? Because we typically measure that by putting the antigen of interest on the plate and then measuring by ELISA whether the antigen specific immunoglobulin goes up. But if you measure total IgG, it depends, but it often will not vary that much. I think if I remember, the IgA in the gut can go up, but I'm not 100% sure about that. But yeah, so I think that usually we're measuring specific antibody responses, but this is more, this is just a total response. Yeah. And so it almost reminds me more of what happens in insects, with the production of antimicrobial peptides in response to infection.
Interesting.
So then they say, what influenza virus proteins are these VLR-Bs directed against? So they take purified proteins from virus particles and they mix them and use an ELISA format. So first they show that these VLR-Bs bind intact virus and not a different virus, a parainfluenza virus, which is of a different family. And then they say if we take the individual proteins of influenza virus, which ones are being bound by these VLR-Bs, and it's over 90% is against the surface glycoproteins of the virus, the hemagglutinin and the neuraminidase, the HA and the NA. And they can also look at virus themselves. They can make reassortants. They could swap out the genes for the surface glycoproteins for different virus entirely, an H3N2, and show that they don't react with that as well. So most of the antibodies are specific for HA and NA, and that, you would see the same thing in mice if you did this experiment in mice. So right away, it's telling you that something is very similar. Here we have this lamprey and a mouse, yet they both seem to be reacting with the same proteins of influenza. The immunodominant proteins, the HA and the NA, are both immunodominant in lampreys as well. And then they take a different influenza virus, H3N2, because who knows, maybe H1N1 is weird. Put in H3N2, you get the same immunodominance. HA is immunodominant. So, so far, it looks like mice and lampreys are giving us the same message. So we make antibodies against the HA and the NA. What are these antibodies doing? Because we know in mice, if you immunize them with influenza virus, the antibodies can have an effect on the virus. The antibodies, of course, could block infection, and they can also block the binding of virus to red blood cells. And that's a common assay for influenza virus, the so-called hemagglutination assay. If you mix influenza virus with red blood cells, they will bind the red blood cells and cause a lattice to form in a plastic well. So if you just put red blood cells in a plastic well, after about 30 minutes, they tumble to the bottom. They form a nice little button. And if you had virus present, they would make a nice little lattice. And I used to do many of these as a PhD student many years ago. And I remember this with great fondness. We used to do this in 96 well plates, and we would have these diluters. So you'd put the virus in the first well, and then we had these sticks that would go into each well. And they had a little head on them, and you would grab them between your two palms and rotate them to mix them. Then you'd pick them all up at once and transfer them to the next set of 12 wells. Mix them again. You'd go all the way down the whole 96 well plate. It was so cool to be able to do that, with skill is very satisfying. Anyway. So what you can then do, if influenza virus causes hemagglutination, you can ask, can an antibody prevent that? Because an antibody directed against the HA may do that, right?
Right.
So that's called hemagglutination inhibition. So if you have hemagglutination, you have a lattice of red blood cells. If you now had an antibody, along with the virus and the red blood cells, you will prevent the formation of a lattice, or you'll have the formation of a button.
Right.
And in fact, they show that these Lamprey VLR-based can inhibit hemagglutination using their H1N1 virus, but not against other H3N2 or influenza B viruses. So mouse and Lamprey VLR-based will have HI, hemagglutination inhibition activity. They will also inhibit viral infectivity. You can infect cells with virus, right? The influenza virus will infect. If you add antibodies, you will block infection. You can measure infection in a variety of ways. You could do a plaque assay. You could do hemagglutination assay. The antibodies to the mouse antibodies and the Lamprey VLRB both can block infection. So far, they're very similar in their biological effects.
Right. They're both there, which is the ELISA, and then they're both functional. Yeah. Which is this test.
So HA is a big target for antibodies, obviously. It's got a globular head, it's got a fibrous stem, and then it's embedded in the membrane of the virus. And people over the years have studied this HA extensively as to what sequences give rise to antibodies. There are lots of antibodies that are directed against epitopes on the head of the hemagglutinin as well as on the stalk. And so what they do here is they use a nice little genetic trick. They have a panel of H1N1 viruses with amino acid substitutions on the 5 defined antigenic sites on the head of the HA. And they simply ask, do these substitutions disrupt the binding of the Lamprey antibodies? That's cool, right? It's a clever way to do it. And they find that, in fact, they see a significant loss of binding with 6 of these substitutions and 60% loss with 12 substitutions. So in other words, the Lamprey antibodies seem to be binding the same antigenic sites on the head of the HA as mouse antibodies.
I thought that was crazy.
Yeah. Isn't that amazing?
I mean, that's amazing.
So basically an antigenic site is an antigenic site.
550 million years separate them. They recognize the exact epitope on the surface of the. And
I guess, well, I don't know, but the question would be, do lampreys ever get infected with influenza virus? Or did they, ancient
right, would they have ever seen it?
Maybe not, right? But if not, it would just say it doesn't matter because any antigen you throw at this lamprey can make VLRs to bind it, right?
Yep.
It's very cool. So they did some competition assays that basically confirm these findings as well. They compete lamprey plasma against mouse, and they can, again, show that the antibodies and the VLRBs are binding to same antigenic site. And they use monoclonals in this case because we have mouse monoclonal antibodies against each antigenic site on the tip of the HA. And they use whole plasma from lampreys. And they can see that some compete and some do not compete. So when you start to break down the antigenic site specificity, it's a little bit different, but that's not surprising because even mice and humans are different in that respect as well.
Right. Isn't the tip of the HA though? It seems to be that's the dominant antigen. So that's what's driving the majority of the immune response. But what we understand though is that that's the most highly variable part.
Correct.
Right. So if what people are trying to do is get more strong responses against the stalk region, which is less variable, right?
Mm-hmm. That's right. The stem is immunosubdominant, but if you can make an antibody against the stalk, it's broadly neutralizing. It neutralizes many different HA types because they're all conserved. The sequences are conserved. And so, that's what people are trying to do is somehow stimulate the production of these stem antibodies. And they do find here, they have one plasma that didn't block binding of an antibody against the stem. And so they say, just like in mammals, the stem is poorly immunogenic in lampreys. And so the lamprey response is focused, just like the mouse response, on the globular domain of the HA.
And I wonder if they thought after they got these results, maybe if we're looking into changing the way that we generate antibodies to something that's not this, to the stalk, maybe if the lampreys were generating it to the stalk, it would be interesting to try to subvert that in some way to find out why that is. But in fact, it's the same.
Yeah,
so it's the peptidic nature of these proteins that's driving the antibody generation and the immunodominance of the antibodies.
So in mice, what you can do is you can do the following experiment. If you infect a mouse with, say, H1N1 influenza virus, okay, and then you go back and boost, you get mostly antibodies against the head. But if you, instead of boosting with the same virus, if you use a different virus where you have the same stem, HA stem, but with a different head, you now get a primary response against the head. You don't get a memory response. And you get a better response against the stem.
Right.
Because now the animal has seen the stem twice with a different head. And apparently if you put a different head, it reduces its immunodominance somehow.
So is that an argument for why we should get our yearly flu vaccine? Because you'll keep boosting against different heads, but the stalk will remain primarily the same. And so you can over time generate more specific antibodies against that, or no?
Well, if you do that, the head, it's a good question, but I think the heads are too similar. If you do year after year, it's really the same subtype, and it's too similar. You have to have a totally different HA. So you'd have to have an H1 and an H4 or an H7 or something that humans have never seen. So that's the Palese approach to it. They're trying that in people, and we'll see if that works. And the idea would be that you only need to do that every, 10 or 15 years, depending on how long the memory lasts. We don't really know how long it would last, right? But they could do that here in lampreys. If they wanted to push this, they could then say, well, can we make a stem response by swapping heads? But of course, we don't need to make lampreys immune to influenza viruses, so we might as well do that.
But they are a small model, and if their concepts translate,
would be interesting,
it would be useful.
Yep.
And that's related. I think my favorite line of the paper was the last one where they said, perhaps when it comes to antibody responses, neither mice nor lamprey lie after all. Because a common thing we say is monkeys exaggerate and mice lie in the terms of the translatability of the science. But I think what this paper argues and shows is that these, like Cindy said, we need small animal models and we can learn a lot from them. So, it's interesting.
Yeah, I like that line. It's good.
Yeah, that's good.
We said it a lot on TWiV. Mice lie and monkeys exaggerate. I wonder if that's where they heard it. Because I know that John Yewdell listens to TWiV. By the way, the lampreys were purchased from a company called Lamprey Services. in Michigan, where I understand that these are an invasive species. So I guess they have people catching them to try and, although I doubt that's going to make much of a difference. I don't know what they do with them when they catch them.
I know we have gobies that they're small fish that have invaded our lake here in Cuba Lake, and if you fish and you catch one, you're supposed to kill it and not put it back.
Oh, wow.
But it doesn't really make much difference because there's so many of them.
Yeah.
I think it's just if you study them. Yeah, Lamprey Services is in Ludington, Michigan. We have all the necessary permits for collecting and exporting. We can guarantee that the species collected is the sea lamprey and that the animals are in good health. The shipment consists of a 48-quart cooler.
Wow.
Fish are in plastic bags containing pure oxygen. They will stay alive for 40 hours. So if you want to work on sea lampreys, that's the page. That's the company.
I guess you could keep them as a pet. I mean, if you were into that.
Are you going to do that, Steph?
I mean, no. I
don't know about that. Isn't there a problem with people keeping those? What are the Asian fish that people keep and then they let them go and they get too big and then they have invasive?
Oh yeah, right.
That
happens often. Alligators in some places, right? You go, you buy a pet one.
So maybe it's not a great idea to keep them.
But that's what has spread the frog fungus around the world. People buying frogs and moving them around, and they have a fungus. They introduce it to a new area where the frogs don't have the fungus, and boom.
Yep.
We really wreck things, don't we? All right.
We do.
Let's do some email.
All right.
Cindy, can you take that first one?
I will try. So Stephen writes, B cells or T cells in TB, a continuing conundrum, the Lancet Respiratory Medicine. Hail to the geniuses who make head or tail of immunology. Here is a Lancet paper I can't even begin to unpick, so I thought perhaps it might be a good one to help illustrate some principles. I'd be particularly interested to know if my BCG scar was all for nothing, as it appears that people can still be infected with TB even after having gone through this scabby procedure as a child. Keep it up, Steve Hawkins, Lutton, Bedfordshire, England. And importantly, he says, "Gray day, but plum blossoms out at last, and birds singing." And I can't do that with a British accent. But I think what's interesting here that needs to be talked about is BCG is Bacillus
Calmette-Guerin.
I never can pronounce that. But it is a less dangerous version of Mycobacterium tuberculosis. And so the thought was, just like vaccines with cowpox for smallpox, as Edward Jenner originally did. The idea is that you take a less dangerous one and it protects you then against the more dangerous one, which is Mycobacterium tuberculosis, which we used to say infects 1/3 of the world, but I think it's now they're saying that's less, but it's still a major problem. But the thing with BCG is it definitely prevents more serious complications. So TB is a respiratory disease that causes large granulomas in the lung. And then if those granulomas stay intact, the individual is okay. Basically, those are walling off of the Mycobacterium bacterium in these granulomas. But if they start to break down, they become what's called caseous, which is cheesy in the middle. And if they bust open, then the bacteria get into the airway and the person coughs, and it's extremely contagious. And so BCG will help the immune system keep that in check. So there are far fewer transmissions of TB and people get less sick from it. The problem is that it doesn't fully prevent the disease. And that's true. One thing that we always get sticky issues about is that vaccines don't prevent you from ever getting exposed to and infected by something, but they help you overcome it so quickly you might not never know you had it. And so and that's true for any vaccine. What we're doing is we're tricking the immune system into thinking it's seen it before, so it can act more rapidly and get things under control more quickly. So even with influenza, if you have the influenza vaccine and it works great, it doesn't mean you won't get sick at all. but you may not know you're sick, or you might get a much less serious version of the disease course. And so the same is true with BCG. So it's not all for nothing, but it doesn't fully prevent the disease. The problem with TB is that this is a long-term thing. This particular bacterium is extremely crafty and has lots of ways to subvert the immune system. And so the primary way to keep it under control is to wall it off. And actually eliminating those bacteria is extremely difficult. So even if you're on multiple drugs, the antibacterial drugs, for a long, long time, and your compliance is good, these people are being treated for 6-9 months or longer of daily hardcore antibiotics. And it's just simply that these bacteria live inside of macrophages, which are immune cells, and they're really good at living inside of them. So if you think about it, macrophages eat bacteria and they put them into these capsules, these phagosomes that are extremely hostile. They're supposed to kill these bacteria. And these bacteria love that environment. They secrete all of these things that just they set it up as a nice home. And so they live within this really, really dangerous and destructive phagosome happily. So it's really, really hard to get rid of these bacteria. So once you get them, they're there for, life, unless you have a really strong, hard course of antibiotics to get rid of these. And so again, coming back to this, BCG treatment or BCG immunization will prevent you from getting really serious complications from disease. Unfortunately, it won't always protect you from ever getting infected. And so you might be TB positive. One of the really interesting things is in the US, we do not use BCG. And we, in fact, use a TB test where you prick the forearm. It's a TB time test to test for TB. And if you've been immunized with BCG, you will test positive on that. And so that's why we don't use it in the U.S., because then you can tell who has TB. And so the problem that we've had in the lab is when people come from other countries and they've been immunized with BCG, they get a TB test. They test positive, and of course, everybody freaks out. Oh, no, now you have to have lung radiographs and everything else to see if you're actually infected with TB, because then they have to worry about what you're working with in the lab and so forth. And often, they're not really infected, but they have been immunized with BCG and they test positive. So there's some interesting biology around all of this.
This the paper you sent is about a new kind of vaccine.
Yes.
And it's actually 4 different proteins with an adjuvant, right?
Yep.
And they make the statement that there are other vaccines. Is it just another vaccine candidate, or is it novel?
There are a lot of vaccine candidates being tried, and I don't know the various levels of success of all of them. Usually they're using drugs to treat it, because trying to get the immune system to overcome it is challenging. So all the vaccines may very well work, but it's getting that getting those immune cells into that granuloma and being able to fight the bacterial infection within the macrophage. How effective that will be is unclear. They have to be tested. So
this is only a phase 1, so we only know safety, and they have a little bit of antibody and T cell information. But really, you have to do a challenge, not a challenge, but you have to send people out into the world and see if they get TB or not, right?
Right.
And of which there's plenty, so that can be done.
There are.
And places where TB in the U.S., I mean, we don't think about it in the U.S. much, but where it can be a problem, where people congregate that are also in high stress situations. So you can think of hospitals, you can think of healthcare facilities like for the elderly, but also, places at the border where people are in high stress and are detained. And so there are definitely some infectious disease implications to how again, when politics is relevant and infectious disease, they intersect.
Right. And so when you have people in those high stress environments, that's when your immune system becomes compromised. And if that happens and that breaks down, that's when TB can get the upper hand and spread.
Right.
All right. Steph, can you take Anthony's email?
Sure. Anthony writes, I heard you mention this on the recent Immune. You seem to indicate that this was an ongoing issue. So I'm just going to preface this by saying I think this was a while back, but we were talking about, using animals, and that pigs as well as mice can savage their young, which evolutionarily, maybe it's an adaptation if there were predators around, or if there was a runt that was sick that could, resources, you're trying to redistribute them. And so I think that's what Anthony is mentioning.
And I had mentioned that our mothers keep eating their offspring, our mice, and we can't get our experiments done.
Right. It's really unfortunate. It's not a fun thing to come upon when you're going in for the day to do chores or whatever. So Anthony says, I raised mice for the pet trade some years back and never encountered this problem. I'd first look at enriching the diet and environment, put the cardboard centers from rolls of toilet paper or sections from paper towels in the cages. This gives the mice tunnels to explore. Particularly for the moms, give them handfuls of paper strips from a shredder to build nests. In addition to mouse chow, he says sections of celery, carrots, chickpeas, whole corn, sunflower seeds, peanuts, apples, whole wheat bread, possibly with peanut butter, and many other items for human consumption can be used. These don't have to be given all at once, just a couple each day. If a conference room generates leftover sandwiches, salads, bagels, these can be collected and used. Hey, graduate students are the ones
they get
those leftovers, not the mice. I'll put my foot down. Don't use avocado or chocolate. So obviously mice have some maybe allergic reactions. Mung bean sprouts are particularly valuable, but these need to be grown on site. The light famished stuff in stores is expensive, nutritionally bereft, and often microbe infested. Well, I guess all things are, but covering the sides of the enclosures that house the pregnant mice with paper can be tried. I've never had to do this myself, but my mouse handling was all kept low. Labs often have mice on high shelves. Mice are not squirrels. Elevation, distressed mice.
That's a good point.
It is a good point.
We do have our cages at all levels, right? Depending on where they are. And, often we also screw them up because they're nocturnal and we put the lights on during the day so people can go in and see, right?
Right. So, and, I appreciate all of these suggestions, but I think, Vincent, you had mentioned everything that goes in has to be sterile and really has to be very controlled. So a lot of these suggestions are good. But, some of these can be applicable, like you said, considering the height levels.
So if I put some carrots in the mouse cage, I would be banned from the mouse facility because everything that goes in has to be sterile in the cage because otherwise you get get infections that you don't want. Now the solution seems to have been we would have litters, and then Amy would go in the next day and they'd be all gone, right? Mm-hmm. They told her, you do the husbandry, you take care of the mice, which means changing the cages and so forth, and that makes a difference because I think the people doing the handling, they'll take the lid off, throw the mice into another cage, right? Mm-hmm. I mean, I don't think they are that's stressful. And Amy is very careful and slow, take one out at a time by the tail. And it seems to make a difference. We now have some litters. So hopefully they'll stay, right?
Sometimes what you can do too is if you're good at knowing when the mice are very pregnant, I guess you're pregnant or not pregnant, but when they're close to deliver, you can put a do not change the cage sign on, and so a little stinky in there, but if you leave the mom for the first couple days, she calms down a little bit, and then it usually doesn't happen. But I want to also respond to Anthony. The mice are quite enriched. At our facility, we do give them, it's not toilet paper rolls, but it's the same idea, these little cage that, these little cardboard things for them to chew on and walk under and snuggle in, and so to tunnel under. And we definitely give them shreddable bedding that they shred up and they make their little home there. So they do get all of that kind of enrichment in their cages, and we try not to house them by themselves because that's also quite stressful. So if you can get a mating pair and leave them together, often they will leave the litter as well. Some people don't like to leave the male in there; it can be bad. But if you leave them together, often they'll continue to mate and have more litters over time.
And how we figured out my pig situation is something that I'm not reinventing any wheel here. It's widely used in the swine industry, but it's something called farrowing crates. And so if you've seen animals in crates on swine farms, I can attest to it's very functional for a functional purpose. And we put them in, at least what we do is 2 days, the anticipated farrowing or giving birth. And then we keep them in there until we can be sure that she's not going to be aggressive or roll on them. And so they are immobilized. They can stand up and sit down, but they cannot turn around, to try to get the baby. So that's the way that both science and the industry has solved that problem.
Cool. All right.
The big pigs will turn around and accidentally roll on their baby.
They're so
they're really big. They
don't know.
It's not their fault.
Especially in a small space like a lab place, right? Whereas maybe in nature it wouldn't happen so frequently.
I mean, that could be true. Although our pigs do have quite big rooms that they're in. So they do have a lot of space. I think it's just right after, they're usually, when they have like 16 to 20 piglets, they're usually giving birth over a course of time. They're standing up, they're going down. So they may be a little disoriented and focus on the birthing and not there's half of the piglets are suckling her and then she's still giving birth. It's a whole,
wow, that's tough.
Whole thing.
My gosh. I have a newfound respect for pigs. All right. Melanie writes, hello to the immune group. I've been enjoying your podcast for a while, Vincent. I'm delighted to listen to your immune series, whereby you have done a great job to explain the basics as well as keep us up to date in a complicated area. I like the format so far. I was looking for an immune mug or shirt to buy, but I couldn't find any. Please direct me in the right direction if there are any available to purchase, or let me know if I need to wait a bit longer. Thank you for making my long commute to work much more enjoyable. I'm at Acadia University in Wolfville, Nova Scotia. Can't wait for the next immune. I was just in Nova Scotia, except I was in Halifax. Very nice. So this morning I made some swag. Is that how you say it? For you, Melanie, cool. I made some mugs and cups and a few T-shirts, a white one and a dark one. You go to cafepress.com/TWiV, cafepress, all one word, .com/TWiV, and you will see there's a whole lot of stuff from all our shows, but you will see immune mugs, the metal things for your water, and a few other things. And if there's something if there's something you don't see that you want, just tell me. I can add. I didn't add everything that's there for the other podcasts, but there's a lot more.
All right, I have to order something. That's awesome.
Thank you for reminding me, Melanie. I had forgotten about that. All right, let's work through the last couple ones here. Cindy, you're next with Brandon.
Okay. Brandon writes, hello, Cindy, Steph, and Vincent. My name is Brandon, and I am an undergraduate at the University of Alaska, Anchorage, working on graduating by next spring. I do research under a professor in the field of immunology and virology, which has led me to be fascinated in immunotherapy for diseases, specifically cancer. I am writing to you to request you do a podcast on the recent cancer vaccine paper. And vaccine is in semi-quotes. I thought your podcast on CAR T therapy was incredibly informative and well produced, and I would find it would be incredible if you guys presented this paper on the podcast. I have included a copy of the paper attached in this email. I love the great content you produced. Thanks. So this is a paper called Eradication of Spontaneous Malignancy by Local Immunotherapy. And I did look this up briefly, and what they're doing and what I like is they're using a TLR9 ligand, which is DNA, to inject directly into the tumor in different situations to induce an immune response. So it's local immunotherapy. And so I think it would be great. They're using this other ligand called OX40. So it's an anti-OX40 antibody plus the CpG DNA, and they seem to see really strong immunity against the tumor and elimination of the tumor. So it's potentially a very exciting new therapy. So we could definitely think about talking about this more on a future podcast.
Okay. Yeah, because our next writer, Bob, sent the same paper in.
Two people sent the same paper, so clearly people are interested in this.
So at some point we'll do it.
And I guess Bob, so if you want to do the same vein there, I can do that. So Bob wrote in about the same paper, and he said, what does the panel make of this? And he sent this. He actually sent a link of a news article about this particular paper, and he quoted something from the paper. It said Levy. So this senior author on this paper is Ronald Levy or Levy. Do you know how you say his name?
I think it's Levy.
I think it's Levy. So Levy's method works to reactivate the cancer-specific T cells by injecting microgram amounts of two agents directly into the tumor site. And so a microgram is 1/1,000,000th of a gram. A short stretch of DNA called a CpG oligonucleotide works with other nearby immune cells to amplify the expression of an activating receptor called OX40 on the surface of the T cells. So CpG-DNA is a TLR9 ligand. The other, an antibody that binds to that OX40, activates the T cells to lead the charge against the cancer cells. Because the two agents are injected directly into the tumor, only the T cells that have infiltrated it, meaning the tumor, are activated. So in effect, these T cells are pre-screened by the body to recognize cancer-specific proteins. So this really is this idea of taking and mixing these two things together, one that's stimulating the expression of this ligand on the T cells, and the other is an antibody targeting that protein on the surface of the T cell. And together, those are really mobilizing those T cells. And because they're doing it specifically inside the tumor, this is a solid tumor, they're inducing those T cells within the tumor to become anti-tumorigenic.
So I guess this would be okay as long as you can inject it, because not all tumors would be
That's right.
amenable to this, right?
So if you have a liquid tumor or an inaccessible tumor, that would not be an option.
If you have lots of tumors like myeloma, right, that you couldn't inject each one.
They did some experiments on metastases in this paper. And I think if you inject into the primary tumor and you generate these T cells do circulate throughout the body. They can leave the tumor and come back into the tumor. And so very well, they might leave the tumor and find the metastases and attack the metastases as well.
Yeah.
And I think with this paper and these two comments, just good to remember. I had a couple of people ask me about this because it was very highly covered in the press. But it was done in mice. And we have talked before what the limitations are with doing experiments in mice and trying to translate that to humans, particularly with these cancer immunotherapies. So I would say go back and listen to Cindy's CAR T cell episode to get the background. And then, we could definitely do another episode because there's a lot of interest in it, for sure.
All right. Steph, you want to take the next one?
Sure. Mark writes, "Hi team, this is a question for you all and possibly a question for listeners of the podcast as well. I was wondering if anyone could recommend some books for people like me who are interested in human immunology, which are not textbooks, something in a casual readable format. A textbook feels more geared to be read with the guidance of a teacher and often assumes the reader has some prerequisite biology study. It's a broad question, I know, but I would settle for a textbook if no one knows anything more casual. Thanks for your time and all you do." And Vincent has a couple recommendations, but I mean, mostly for me, it's been textbooks. There hasn't been a lot of, I don't think there's as much in immunology as there has been with the pathogens, viruses, bacteria. Because I think there's more, not shock value. I don't want to say that, but there's more interest gearing, and immunology, I don't know if casually can be coined compared with that.
So I like Lauren Sompirak. He has a series of books on viruses, bacteria, and he has one called How the Immune System Works. And these are very good. The virus ones are excellent. He has a knack at explaining things in a simple fashion. And so there's one for the immune system. So I would recommend that. I'll put a link for that one. And Cindy, you had one too, right?
So it's a book I read a number of years ago, but I thought it was really fascinating. It was called A Commotion in the Blood by Stephen S. Hall. And I found it great because it talked about Coley. And Coley was a physician about 100 years ago who was trying to treat cancer. And actually, he was treating one of the Rockefellers who had a bone cancer, I think, with tuberculosis extracts. And it turns out that we now know one of the active components in that was CpG-DNA, which is the TLR9 ligand. And Coley Pharmaceutical was begun based on that. But it has a lot of really interesting information about the historical overview of how we've tried to harness the immune system basically for what we call now immunotherapy. But in that time, it was the anecdotal observation that people who got really sick when they had cancer would often or would sometimes recover from their cancer. And so what was it about infection and cancer? And now that we know a lot more and we've broken this apart, it's probably having to do with being able to break the tolerance to the tumor and the immune and, overcoming the immune system. Everything that we're looking with checkpoint inhibitors and activation of T cells, what we just talked about within the tumor, is all geared towards that. The tumor is good at shutting down the immune response. And if you can overcome that, you could eliminate the tumor. And so the idea was if you got deathly ill with something that was infectious, maybe it might stimulate break this tolerance in the tumor. But it generally talks about immunotherapy. It wasn't called that when this book was written, but the historical overview of how we've harnessed the immune system. And so, I think it comes back to the idea, though, that there aren't a lot of books on there on sort of the overall general, what is the immune system and how it works. But there's more specific books on various different aspects of the immune response.
Maybe you need to write it, Cindy.
In all my free time.
Yeah. Okay.
I know if you like vaccines a lot, Paul Offit has several books that are very, very good on that as well, including a new one that's come out. So it just depends on your, the types of things you're interested in. My colleague down the hall also wrote one on microbiome called the Human Superorganism, by Rodney Dietert. I spelled that wrong in the show notes. But so you've got these ideas. If you're interested in microbiome, there are several books about that. If you're interested in vaccines, there's books about that. Cancer and cancer immunotherapy, there's books about that. But sort of this general, how does it work is not very good. There are a lot of YouTube videos that can teach you a lot.
One more is from Nathan. I'm loving the Immune Podcast. Keep up the great work in your podcasting and tweeting. I recently found this 2017 review that touches on both immunology and viruses. What a great name, super antibodies. Plain ordinary antibodies are quite amazing. Some might even say super. I thought it would be a great topic for either a snippet or full discussion. It is 29C, 85F, and partly cloudy in Chapel Hill. So Nathan sent a link to a review article in Nature Reviews Immunology. It's called Passive Immunotherapy of Viral Infections. Super Antibodies Enter the Fray by Laura Walker and Dennis Burton. And this is about broadly neutralizing antibodies. And they have now been found for multiple viruses, HIV, for example, influenza virus. And we talked about it for respiratory syncytial virus not too long ago. So it's a nice summary of how these are found, by cloning B cells from people who have had these infections and identifying rare ones that are broadly neutralizing. But we have actually talked about this concept before in the RSV episode.
Mm-hmm. And really the only thing that makes them super is that they're broadly neutralizing, right? I mean, there's not anything specifically different about them structurally.
No, they're just broadly neutralizing. We didn't know about them until we were able to, because they're so rare that you don't find them unless you can clone individual B cells. I mean, that's really the technology that enabled this, right? You pull out a single B cell, and then you can clone out the mRNA encoding the antibody. Remember, the B cell is making one antibody, right?
Yep. in large amounts,
and then say, look, it's broadly neutralizing. It's amazing technology.
Yeah, and the idea of passive immunotherapy, just to put that in perspective, is that's taking, like what you just said, taking an antibody that either someone else made and you purify it, or cloning it and making it synthetically in a pharmaceutical company. And passive means that you are yourself not making it, but you're getting it from outside.
That's right.
So if you have the viral infection, you get the injection of the antibodies to treat the infection.
So you have two approaches. You could give people these antibodies, or you could figure out a way to induce them, as we spoke earlier about for flu. That's a little harder.
Right.
Steph, I'm going to ask you a hard question.
Okay, I'm ready.
Would you need broadly neutralizing antibodies to treat Ebola patients?
Well, no, because currently there's only one strain of Ebola, right? So it doesn't mutate like HIV.
Perfect. Great answer. You got an A.
Do I pass?
You got an
ding,
A.
ding, ding.
Nice job. You must have been listening.
Graduate.
You must be listening to a podcast.
I do listen to a lot of micro TV. This is true.
I'm sorry to put you on the spot. I knew you would.
No, it's good. You know what? It's good practice for a couple months when I have to do that.
My son had an interview recently where he said one of the questions was a trick question. It's kind of like what I just gave you. Do you know this thing called ping? If you ping a website, there's a command you can do, ping and then your website, and it will tell you if it's running, basically. And so the person asked him, what port does ping run on? He said, it doesn't run on any port. It's a trick question to ask something like that, right? Same thing.
Well, I'm glad I passed.
Let's do some picks of the week. Cindy, what do you have for us?
So there was a whole media frenzy about a week and a half ago, and you probably know more about this than I do because you're a polio researcher, is they thought that in Venezuela we were detecting a new case of polio. And then they thought maybe it was a vaccine-induced case, which I started to get panicked about because I thought that would be terrible if that actually happened. But it turns out that they looked into it, and in fact, it was not polio. So it was a case of flaccid paralysis, acute flaccid paralysis in a child, 34-month-old child. And so that is a strong indication of something like polio. And so they isolated the polio virus from stool, and then, all of a sudden they said, well, that must be the reason. But it turns out that this individual did not actually have polio or wild polio or vaccine-induced polio.
So this is a way that WHO screens for polio, right? They look for kids with acute flaccid paralysis, which means your leg muscles are flaccid. They are paralyzed, and it's enough to be able to see this. And they go from village to village in many parts of the world and do this. And if they see a kid with AFP, which can be caused by a lot of things, not just by polio, they then take some stool and look for polio virus. And what happened here is they found polio virus in the stool. And so, oh my gosh, and that's when the press came in. And even I tweeted last week, case of polio in Venezuela, which is terrible because it's been declared eradicated there for many, many years, right? The Americas are polio-free.
That's right.
But they did isolate poliovirus type 3 from the stool. So why do we know that this did not cause the child's disease? That's the question. That's a hard question. I wouldn't expect many people to know the answer.
So they sequenced it, right?
Sequenced it, and they saw that it is vaccine type 3, but type 3 can cause vaccine-associated disease. So how do we know that that virus did not cause the child's polio? And that's because it has to have a certain number of mutations in the genome to be able to cause polio. And this isolate looked pretty much like the vaccine strain. There had been actually a mass immunization campaign in the area just shortly before. So type 3 and type 1 were being used. They were being given to kids, oral polio vaccine. And so this child was in an area with declining vaccine coverage. It's gone down to about 60% now because there's no polio. And they say, who needs the vaccine, right?
That's scary.
So the kid picked up the circulating vaccine-derived virus. When they sequenced it, they saw that it was very close to the actual vaccine, and so it was not likely that it caused any paralysis. Now, interestingly, these viruses will circulate in the population for many years, these vaccine viruses. And each year, the genome changes about 1%. And so you can actually calculate how long these viruses have been circulating by sequencing the genomes of stool isolates. You can say it's been around 5 years, 10 years, and so forth. And as it circulates, it picks up more and more mutations that make it likely to cause paralysis. And interestingly, that is what has caused most of the polio last year, 2017, in the world. vaccine-derived polioviruses. In fact, in Syria, there were something like 70 cases, all type 2 vaccine-derived. And that's why WHO is slowly moving away from the infectious, the OPV, the Sabin vaccine, because it causes polio. You got to switch to the inactivated vaccine. But unfortunately, that has to be injected. It's a lot harder. Got logistical issues and so forth. So that's the
I should have put on here too, though, because wasn't there something that just came out where the Gates Foundation challenged people to work on solutions to be able to keep the cold chain for vaccines like the inactivated polio vaccine, so that you could distribute them worldwide? And they came up with 2 really cool solutions that I haven't looked into in great detail, but it's suggesting that maybe we can do this switch over to the safer version of the polio vaccine, so there aren't any vaccine associated.
Well, we have to make it. Well, both vaccines have to be frozen. Both the Sabin and the Salk both have to be frozen. But the real thing with the Salk is that it has to be injected.
That's right,
and that means you need needles, and you need a person who knows how to use a needle. Because OPV, you just feed it to the kid, and anybody can do that, right?
You can put it on a sugar cube, right?
So what the Gates has done, though, is there are 2 things that are worth mentioning. One is that they're putting money into testing microneedle patches, which are little patches with very tiny needles that you put on your skin with a Band-Aid. It delivers the antigen to the skin. They're really immunogenic. It's been tested with polio vaccine, inactivated polio vaccine. It looks really good. So that could get around that, but that's going to be a few years off as well. The other problem is we have this Sabin vaccine, which is infectious. It's really good at giving you gut immunity, mucosal immunity, and that stops outbreaks. That's why if there's an outbreak in Syria, we go in with Sabin because it stops the outbreak. IPV is not very good at that.
Right.
We need a new vaccine that is as immunogenic in the mucosa, in the gut mucosa, but doesn't revert. And the Gates has supported some interesting research, mostly in the UK, where they have made changes in the viral genome to prevent reversion. And these are actually being tested in people. And if they are immunogenic and give rise to the right antibodies and so forth, they would be used post-eradication, because if we can eradicate polio one day, but let's say it comes back, right? And there's an outbreak. What we're going to do is go in with OPV, and then we're going to seed the environment with vaccine-derived strains again. And that's not good. We need something that's not going to revert, but will be just as good at stopping outbreaks. So that's the story there. I'm sorry to talk so much about it.
No, I wanted you to talk because you know more about it, but I just thought it was a really important thing to talk about, because it just goes to show you that the news went crazy about this, there's now polio, and then, it's vaccine-induced polio disease. And then now we find out that that's really not the case. So reserving judgment until all the information is in is sometimes wise.
But vaccine-induced disease itself is very common.
Yeah.
If you go to polioeradication.org and then you go to Polio Now, you will see the number of cases, and they're all vast majority of. So last year there were at this point last year, which is June, there were 5 wild polio cases and 6 vaccine-derived polio cases. So it's happening because we use this vaccine that's prone to reversion. It's known, and it's a problem with it that we've known from day one. It's why the U.S. stopped using it in 2000, because there were 10 cases of polio a year in the U.S., all caused by the vaccine. It was unacceptable.
Right.
So now, it's being used extensively globally, so we do have vaccine-derived disease. We have to get away from that, and that's hard. So that's the story. Yeah, I even tweeted that out a couple of weeks ago, because I thought, oh my gosh, this is why we can't stop vaccinating after we eradicate. How can you stop vaccinating when this thing could pop up anywhere, right?
Yep.
But I'm glad it's not really polio.
Yeah.
All right. That's good. Thank you, Cindy. Steph, what do you have?
So I have 2 TWiV episodes that I'm going to cross promote here because they are on the author, the co-corresponding author on the paper we just did, John Yewdell. And the first one I think is from 2002. It's episode 208. And the second one is more recently, where he goes on with his son.
Both that was really a great one.
Yeah. Both are really good. I will say, if you're in biomedical science and you're in training like myself, it's not for the lighthearted. And I appreciate John's ability to talk about the realities of science and science education in the biomedical field and how postdocs have seemingly turned into 2 years to 5 years and people having a hard time finding jobs. There's a lot of issues, and I think he speaks very truthfully about the problem. So I would recommend those two TWiVs for your listening ears.
Yeah, he's a good speaker. He was very good on those.
Yeah.
That's cool. Thank you.
Sure.
All right, I have an article at Science-Based Medicine, which is a wonderful blog, by the way, written by mostly physicians, I believe. This one was written by Stephen Novella. It's called "Is Gaming Addiction a Thing?" You may know that the World Health Organization has just added a gaming disorder to their official list of health conditions in what they call the International Classification of Diseases. So
I saw that. Yep.
Gaming addiction. And so this article goes through it, and they say, what is it? Is it really an addiction? What is an addiction, and how does this relate? And so really good, basically. And in the end, Novella says, yeah, it is an addiction because compulsive behavior is an addiction, right? And this is part of it. And he ends up by saying the best part is, humans to an extent are not much different than rats. We have a reward system wired into our brains. When we pull the lever and get a reward, something that gives us a pleasant shot of dopamine, we pull the lever again and again. We just need to know when to stop pulling the lever. For some people, it's easier said than done, and they may need some help taking their hand off the lever.
Or the phone or the controller.
Yeah, that mechanism, I mean, that's really why scroll feeds, your timelines you can scroll on your phone because every time it's the anticipation of something new. And there's little shots of dopamine. You continue scrolling and you look at the clock and 25 minutes have gone by. So yeah, it's definitely a thing.
It is. And in fact, Novella admits to being an avid video gamer.
Well, yeah.
I don't actually play video games, but I have other addictions like podcasting.
I think the important thing I took away when I looked at some of the stuff about this was that the WHO was very careful to say this is not just, something that everybody has and they want to look at their phone, that this is very specific. It's debilitating. It's interfering with your daily life. It's ongoing long term. And I think was saying it was going to apply to less than like 2% or some low percentage of all people would fall into this classification. So it's not something, as much as I would love to, they should throw around with your child saying, you have this disorder, we have to do something about it. But so it still doesn't take away from the fact that those types of things are compulsive behaviors, and they do give you the reward and the dopamine shot that you want to go back and look at it.
Yep.
Yeah.
All three of our kids game all the time. And my, I don't know if both of you know this, but there's a whole industry out there. People play games and other people watch them on the internet.
Oh yeah,
right?
Drives me nuts. No, my kids do this. They spend half their.
Do they go to Twitch? Do they go to Twitch? That's where
They do some other things, but yeah, Twitch is one of them.
So my son told me the other day, there's a guy on Twitch who makes $500,000 a month.
Yep.
Because people subscribe to him.
They sit 18 hours a day and play video games.
Wow.
Well, they have food delivered to them.
If you paid me $500,000 a month, I'd do that. Yeah. Hell, I'd podcast 18 hours a day. Nobody would watch.
There was an article in the New Yorker about that, and these people, they have managers and they get promotional people who sponsor them, and they end up very unhealthy because they're sitting in a chair for 18 hours a day in the same spot.
Right.
Well, if you guys want to make $500,000 a month podcasting, let me know. You'd have to.
While that would not be a bad thing.
You'd have to give up your other jobs. We'd all have to give it up, but that's amazing.
I might be okay with that, if we do a couple of months and that's it.
You don't need. You need a year. You get a year and you can retire, right? Or you can fund your lab or whatever. All right, that's Immune number 9. You can find it at Apple Podcasts, microbe.tv/immune. If you have a phone, that you're addicted to or a tablet. On it is a program or an app that you use to listen to podcasts, and you can just search for Immune and subscribe, because you'll get every episode as we release them. There's only one a month, so it's not going to clog up your phone, and we'll appreciate having a subscriber. It helps us a lot. And if you really like what we do, consider supporting us financially. Go to microbe.tv/contribute. Today's episode was presented by Cindy Leifer, who's at Cornell University. Thank you, Cindy.
Thank you.
Steph Langel from Ohio State University. Thank you, Steph.
Thank you.
Stephanie is Stephanie Langel on Twitter, and Cindy is Cindy Leifer on Twitter. And I'm Vincent Racaniello. You can find me at virology.ws or PROFVRR on Twitter. Music on Immune is by Steve Neal. His work is at stevenealpercussion.com. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.